but attending this course? Am I audible to you?
Yes, madam, you are audible, madam.
Okay, so what is the background of the people who are attending this course?
All are almost belong to the biotechnology and science group only, madam. One of them
Okay. Yes, is there any candidate who is not having biotechnology?
no, madam.
Okay, then fine. So when I think you must be knowing that when paddy is getting cultivated, this paddy has got that rice straw has got a very good amount of silica. And do you know that where from that silica is coming to the paddy straw? It is coming from the soil. And when we are cutting this paddy from the soil and we are taking it out, the soil, the silica concentration of the soil is gradually getting decreased. So what the people they are telling that Podali and Podali burning, this is a very acute problem in Punjab, Haryana, Delhi and adjacent places. So what they are doing, they are burning this paddy in the field. As a result, when 1 ton of paddy is rice straw is getting burned in the soil, so after harvesting the rice or the paddy, they just burnt the rice straw in the soil, which is accumulating the silica. Once again, the soil is fixing that silica and other nutrient in the soil in the form of ash. It is getting deposited on the soil. So when we are just burning 1 ton of rice straw, it is producing 199 kgs of particulate matter, 1,460 kgs of carbon dioxide, 60 kgs of carbon monoxide, 2 kgs of sulfur dioxide, and 199 kgs of ash, and 3 kgs of particulate matter. So once we see the burning of rice straw, 1 ton rice straw is giving 3 kgs of particulate matter, 199 kgs of ash, 1,460 kgs and 2 kgs of sulfur dioxide. Just imagine that how much is released to the atmosphere, the environment, and that is creating a huge health hazard to the people of northern part of India. Similarly, when we are talking about, and just it is worth mentioning that it is not that 1 ton of rice straw is getting burned. Millions and millions of tons of rice straw is getting burned. So just you can imagine that how much is the accumulation of this particular pollutant in the air. Similarly, when we are talking about this water pollution, then we are just talking about this. Here's some of the pictures of which are there. It is not only polluting the water bodies, it is causing that marine dumping, industrial waste, sewage, and mainly the household, nuclear waste, oil pollution, and so on. So that is creating the lakes, the rivers, ocean, aquifer, and the groundwater totally polluted. And when it comes in contact with a living being like us, we are also directly and indirectly getting affected with this. So this is all about the environmental pollutants. So if we now want to characterize these environmental pollutants, then this pollutant can be classified as source, that is the point, or non-point pollutants. So these are some of the examples for non-point. So examples are lying with urban, rural, cropland, animal feedstock, and these are the examples of non-point sources of pollutants. And when we are talking about the point sources, that means we are talking about the wastewater treatment and some effluent which are coming from the industry and they are getting treated, and that those pollutants are the point pollutants. So this way we are just classifying the point and non-point pollutants. Every year more than 8 lakhs people around the world die from waterborne diseases, and it is we who are directly and indirectly responsible for this. It is not that only pollutants are getting released to the water body. As a result, the heavy metal in the water bodies are getting increased as for which the mercury in position minimata, Iti, that is cadmium accumulation, which from the contaminated rice is causing some of the diseases, and these diseases are the remarkable disease and identifiable disease. And when we find that yes, this type of people are suffering from this type of diseases, we immediately identify that yes, these are the pollutants. Besides this, arsenic, nitrate in stomach partly get changed into nitrite, which can produce cancer in the stomach. Fluoride, this is called when once it is getting this thing, it is affecting. I think you must have seen that in your toothpaste, some amount of fluoride is there. So if it is in excess amount, it is causing fluorosis. Pesticides in drinking water are also creating health hazard, starting from DDT, Altrine, and some other such type of pollutants. So once we are going for the pollution generation, that pollution generation sources include domestic to agricultural sectors, where hospital, municipalities, industrial, institutional, those everything includes there. And once we are further categorizing these particular pollutants, we are categorizing as organic, inorganic, and some of the pathogenic hazardous waste, which are having direct impact on health hazard. So if we decide that in which way, what is my topic, that in which way we should go for the treatment of this type of pollutants, then we can find that the entire stream can be divided into organic and inorganic. So if it is we who are deciding that what will be our treatment stream. So if this is the mostly the inorganic, we generally prefer incineration, pyrolysis, plasma pyrolysis, or vitrification, encapsulation, gasification, reuse derived fuel, and so on. These are the techniques. But when we are going for the organic treatment, it is we who can go for aerobic treatment, who can go for anaerobic treatment, who can go for directly burning of the organics to this pyrolysis. These are all chemical processes that can be adopted for the treatment of this. But on the contrary, that we can go for the biological or biotechnological intervention for the organic waste which are getting produced, and sometimes we are taking the hybrid process, that is the mixture between aerobic and anaerobic, instead of burning them thermally. And that may be the reason why I'm just talking about composting, farmyard manure, vermicompost, anaerobic digester, and along with that we are going for some fruitful product formation, and that fruitful product may be any high value, and that may create some revenue and waste to wealth concept has come from this particular treatment processes, where it is decided whether we can go for any waste to wealth things. So here the incineration process, so here we are burning it in a very high temperature, and there are plenty of advantages and disadvantages. So these are the incineration plant which are generally operated at very high temperature. This is pyrolysis. When pyrolysis is taking place, we generally go for the pyrolysis process, where irreversible chemical changes are brought to the action by the heat application, and this process is not that high like 800 to 900, but it is 200 to 300 degrees centigrade, but in absence of oxygen, and we are getting the liquid product in the form of char, and that is the process. Here different types of pyrolysis processes are there, and we are getting obtaining either char, which is once again getting storage, we are getting syngas, we are getting bio oil, and so on. This particular processes also can be utilized for the electricity and heat generation. So this is the pyrolysis. These are all chemical processes, and this way that plasma pyrolysis, vitrification, and gasification processes are the standard protocol, where this particular waste material is subjected to a particular temperature depending upon the what type of waste we are considering for the treatment process. So in case of gasification process, we are subjecting at high temperature, and this temperature is at 650 degrees centigrade. So this is some of the waste gasification plant, and we are going for the product process development. This is refuse derived fuel and encapsulation. So here some of the treatment where we are going for micro encapsulation process, or sometimes we are going for the macro encapsulation process. So when we are going for the chemical treatment processes, those are all chemical. So you can find that this system is an energy driven process. So here many a time many of the products are leading to some of the intermediates, and those are very, very toxic and not at all preferred. So preferred reactions. So in that case, what is the alternative? And now when we are going for these treatment processes and its alternative treatment of the pollutants and its different techniques, we are generally talking about the biotechnological intervention. Now, obviously, the question comes that why environmental biotechnology? Now, environmental biotechnology can solve the environmental problem, the problem associated with the environment, with the proper application and selection of the processes for that particular type of pollutants which we have considered. Now here I want to mention it categorically that we have considered organic pollutants for our particular purpose. We are not mostly, the inorganic pollutants are not mostly preferred. And here, when we are going for the organic pollutants, we are talking about the different types of treatment which we look forward for this. So when we are going for this biotechnological treatment process, obviously the terminologies like BOD, COD, and so on is coming into the picture. The next question, as this is a very mild process, it is not producing much intermediates, or the intermediates which are getting produced can further be utilized for some other processing purposes. people are now very much looking forward for the biotechnological intervention for controlling the pollutants. And that is the reason why BOD, COD like terminologies have come into the picture. There are certain relation between BOD and COD. So when we are just talking about this environmental pollution and the biotechnological remediation, we talks about inorganic and organic. Inorganics are not mostly preferred for this biotechnological pollutants, and organics are the most preferred processes where solid, liquid and gaseous pollutants can also be getting produced. So everything you just see in triple form is there. In case of environment, we talk about soil, water and air. In case of organisms, we talk about microorganisms, we talk about plant, we talk about the enzymatic degradation or enzymatic intervention for the organic pollutants which can be taken care of. So when we are talking of this mechanism of bioremediation, here this pictorial form, this is the most beautiful and most preferred slide of mine. So here you just see that number 1, 6 and 8, it is the phytoremediation. So different forms of remediation of the pollutants. So one is this, 6 is this, 5 and this is 7. Where 6 has gone? 4, 5, 6 is here. You see that 1, 6 and 8. These three are the phytoremedial pollution controlling, whereas the microbial tools which are there, the microorganisms which are intervening the pollution control problems are the 2 and 7. So here you just see here this 2 and this is the 7. Here these microorganisms are also there. That means when the water bodies are there, and here this microorganism that naturally when it is intervening the process. So 3 and 3 is the enzymatic conversion of this some of the pollutants to some non-hazardous intermediate which are there. And 5 and 7, this is the 5. Here you can see some of the Bhanmi things are there, and 7 is this. So these are some of the that is zoo remediation, zooplankton, phytoplankton, and so many things you have heard, along with the phytoremediation which are there, are taking part in this particular system. Automatically the nature is taking care of the particular pollutant with its own way, own manner, and that may be the reason why the nature is taking a very long, sometimes a very long time. Say for example, if the pollutant is in the form of tannin, it takes a long time to degrade that to the natural or remedial non-pollutants type of intermediate. And this is the overall mechanism for biodegradation, where in one hand the plant or phytoplanktons, zooplanktons and microorganisms are synergistically working together with controlling some of the pollutants which are present. Along with that, some secreted enzymes by the microbes which are there in that particular system is also converting or also helping to convert some of the intermediate to some toxic material to some non-toxic intermediates. So this, if this is overall strategy, then how one can go for the different types of or different steps of biodegradation studies. So the people like you can go for the biodegradable studies by isolating the microorganisms in the laboratory. Then you can go for purification, then identification of the microbes, optimization of biodegradation product, determination of the intermediates or the end products which are getting produced from this, identification of the biodegradation product, cell or enzyme immobilization for faster degradation of this, and then you can go for the identification of the enzyme, that how much it has taken part for conversion, how much it is entrapped, or is there any reorientation, and that way we can go for the studies, systematic studies on biodegradation of the pollutants which are there in the environment. So the fundamental cleanup reactions mainly takes place with the microbial intervention. Besides that, some oxidation reduction reactions are also taking place in this particular reaction when we go for aerobic and anaerobic system. So when we go for the aerobic and anaerobic, the system itself talks about the presence of oxygen and absence of oxygen. That means oxidoreductase type of oxidation reduction types of reactions are there when we are choosing the aerobic as well as anaerobic system for the biodegradation studies. So the players in metabolic engineering, when we are talking about the microbes, so naturally we can find that yes, there are different metabolic pathways, metabolic intermediates which are there, are playing a significant role, and from this metabolomics we can get a real picture of the conversion of one product to another. So say for example, if we have taken the most complex product like petroleum, so it starts with the benzene to some other component which are there, and it is having some impact, some pattern, some pathways that it is follows for cleaning those pollution, and that cleaning up is mostly done by some of the bacteria, that is hyperactive bacteria, and the most prevalent in case of the petroleum industries, this product is that a microorganism which is predominating is a Pseudomonas, different strains of Pseudomonas. Besides that, there are several types of fungi or algae. They are also participating in this particular process. So when we are going for the bioremediation of this genomics pattern, then we are going for the stimulation of this bioremediation process. And by looking at that stimulants or added of that or the pattern of those stimulants, we can go for a huge in silico studies of this particular remedial method for the computational programming and analysis, data analytics, and so on, the recent development with this particular data on which we are getting this information. Besides this, adding bacteria or fungi to the system, it is helping or fastening the process, and that process sometimes is known as bioaugmentation or the seeding process. So when we are going for this type of pattern, this type of studies, in silico studies, from there with the good modeling and simulation process, simulation studies, one can get the overall picture in such a beautiful manner. Sometimes the expected and predicted data are very close to each other, and from that pattern one can have, by not doing all experiment at a time, but by doing the in silico studies, they can predict that which condition is the best condition for taking that type of pollutant into consideration, and which type of microorganisms can be of maximum use or useful for that particular process. So when we are going for this type of high level research related activities for this very minimum priced of our environment, creating health hazard, this type of pollutants. So I'm just showing you that how the orientation is taking place, that low cost product is now becoming very high cost product, and we are changing this thing to the value addition, and we are going for the high value product formation. And that way what we are telling is that that when we are just going for this type of studies, sometimes we are considering some of the microorganisms like wholesale, or sometimes we are considering some of the microorganisms, some of the enzymes, and those enzymes are getting immobilized, and there are certain cross-linker or techniques which can also be followed like any other biotechnological activities in this waste management or waste treatment process also, or biotechnological processes of waste handling. We can go for this type of carrier binding method, entrapping method, and cross-linking method, and these methods can be used equally for this type of product process handling and intermediate. Many a time the material which are used for this particular purposes are polysaccharides, some proteins, some synthetic polymers. Sometimes it is in the form of inorganic material like glass, sand, ceramics, and some are magnetic. So while coming to this phytoremediation, I think many of you have heard of this hydroponics. Have you heard of this hydroponics? So here this phytoremediation is the some of the examples I'm just giving. This phytoremediation is used in plant for accumulation, removal, and conversion of the pollutants. So when we are talking about this phytoremediation, it is first, it is going phytostabilization, it is going for phytostimulation, it is going for phytoextraction, and it is going for phytotransformation, and overall it is the volatilization. Now, this is the very good example of this phytoremediation of the pollutants. You see, these pollutants are there. So first it is coming to the root zone, and this is getting stabilized. So once this stabilization is there, so phytostimulants are there. So phytostimulants are taking care of this particular pollutants. So when some pollutants are going to the upper part, so it is getting accumulated either in leaves or any other part of the system. So when it is getting accumulated in the suppose in the leaf of the particular plant, then it is getting extracted, and it is once again coming back to the soil. And when it is sometimes it is getting the other technique, So besides this extraction, if it is not getting extracted, these pollutants are there, and through the stomatal opening and the different metabolic cycles, sometimes it is getting volatilized and coming out of the plant bodies. And sometimes this particular pollutants are getting degraded. And this is the overall mechanism of phytoremediation. So this is the some example of this, the guided things of this phytoremediation. And here artificial wetland is getting, so I was just talking about the hydroponics. So hydroponic is one of the example of this phytoremediation, and there are, besides that, there are more than 400 species of plant which are coming for this hyper accumulation of the heavy metals, such as some example are grasses, sunflower, corn, hemp, flax, alfalfa, tobacco, willow, Indian mustard, poplar, and so on. So these are some of the examples which can, so that way 400 plant species have already been classified, which can accumulate it, that they are the hyper accumulator of those heavy metals which are there in the water bodies. So when they are just going to that particular thing, the root extrudes of those plants may be an important role for phytoremediation, and it activates the surrounding microorganisms, and microorganism is then becoming very active to take care of that. Sometimes genetically engineering techniques are used for Bt protein or insect that is called pheromone producing plants to reduce the uses of pesticides to the soil. So these are some of the techniques which are there. So phytoremediations are for these heavy metals are there in different techniques. So the metals are getting immobilized or it is getting solubilized. So once it is getting solubilized, means it is bioleaching process is on. If it is immobilized, then it forms the complexation or precipitation, and that way it is also getting separated from the contamination. So these are some of the examples. The biosurfactant, I think you must be knowing, biosurfactants are some of the chemicals of which has got the hydrophobic as well as hydrophilic, the polar and non-polar resin, and that is why some of the molecules are called amphipathic in nature. So these are some of the examples that biosurfactants can be produced from some bacteria, cyanobacteria, fungi, yeast, and it is classified into different categories. So when we are just going for the physiological role of biosurfactant, then we can find that increasing the bioavailability of the hydrophobic compound, nutrient storage molecule, it saves the microbial cell, this effluent for this harmful compound, and extracellular and intracellular interaction as the quorum sensing and biofilm. So these are some of the components which are very, very costly, and it has got plenty application in other biotechnological applications. So these are some of the examples of biosurfactant. So these bioremediation techniques can be in situ and ex situ. That means without excavation or with excavation. So when we are going for the in situ process, it allows the efficient optimization, and for that this particular biostimulation is dependent upon different factors, environmental factors, which are very much dependent on remediation techniques. So here you just see, this is being the person of agricultural and food engineering department. I feel very much comfortable to tell that how land and water systems are getting polluted with the application of chemical pesticide, herbicides, insecticides, and so on. And here how bioremediation can be taken place with this particular system. So these are some of the examples of the different types of pollutants. In our laboratory, we have worked on different types of phenolic compounds which are very difficult to degrade. And 2,4-DCP is one of such examples where we have taken this particular example 2,4-DCP through enzymatic means under biological treatment process, and we have treated this 2,4-DCP into the fruitful, neutral, and the harmless product, which will not have any drastic impact on the environment. For that, we have used the enzyme called lacase, and this lacase was efficiently removed more than 98% of this 2,4-DCP within a very short span of time. Within few hours, it removed that thing. So if we are really very much enthusiastic and serious about abating the environmental pollution, these are some of the examples. From it we can take a very good lesson, where the Exxon Valdez oil spillage were there, and as a result huge living system got affected. And another thing in oil field in Kuwait has also created the environmental, that oil spillage in water bodies. And whatever may be the thing, if water is getting contaminated, then entire ecological system are getting disbalanced. And with the pilot plant studies, these remediations were possible, where it has the bacterial or microorganisms have taken a proactive role to remediate these pollutants which are there. And pilot plant studies was also conducted to treat the chrome sludge generating from electroplating industries and some of the microorganisms which are already there. So pilot scale studies in different refineries in India is already been practiced nowadays for petroleum hydrocarbon pollutant remediation, and there the bacterial consortium is playing a significant role for this type of pollutants to be taken care of. And once I'm just informing you that this is the present scenario which is being practiced, and in everywhere, whether it is the oil industry, whether it is anything and everything, you will be finding that until and unless you have the environmental clearance, you cannot open any industry, and the pollutants which are getting generated are to be taken care of by the different means. And biotechnological intervention is one of the important area of pollution abatement. And I hope with this small 1 hour information, you will have that much idea that yes, in which way the world is going. So starting from pollutants to the oil spillage and the petroleum industry, how they are treating the pollutants generated in their factory through biotechnological means. Hope you have understood this particular the concept, and I wanted to tell the latest updates of this pollution abatement. And I started from the very preliminary, and I think I could able to give you an overall picture that in which way one should go and how one can take the decision for pollution abatement. So thank you very much. I think I have taken 5 minutes extra. Okay, thank you very much. Okay.
No, madam, exact 11, madam.
Is it? Okay. So in my watch it is 11:05. So I'm really so that is why hurriedly. So if there is any question, any queries, you please tell the students to tell. I will happy to be to answer them. Okay.
Dear participants, any queries? I think everything is clear and very informative,
Yeah.
madam.
Thank you. Thank you very much. Thank you. Okay.
Thank you very much, madam, for delivering the lecture on environment pollution and relation with biotechnology. Thank you, madam.
Thank you, thank you. So I think you are now satisfied with this presentation, because yesterday you were very tensed. So when I got confused with that September and October, I thought that it is in October. So that is why I was not ready with this presentation. So that is why. So,
Thank you, madam. Thank you so much.
thank you very much.
So, dear participants, let us take a short break and we'll join at 11 a.m. again. 11:10. I'm sorry, 11:10. I have posted the feedback link for the session 1 of today's seminar. I request all of you to fill it. Thank you. Let us meet at 11:10. Thank you.
చందన గారు వినిపిస్తోందండి?
మేడం వినిపిస్తుంది మేడం.
కనిపిస్తోందా స్క్రీన్?
స్క్రీన్ కూడా కనిపిస్తుంది మేడమా?
ఓకేనండి, ఇది ఇలా చేసి ఉంచేనా నేను?
ఓకే, స్క్రీన్ కనిపిస్తుంది, మాట వినిపిస్తుంది.
థ్యాంక్ యూ సార్. అదే ఒకసారి మీరు చెక్ చేసుకుంటారన్నారని మేడం ఆన్ చేశానండి. అంటే నేను వీడియో ప్రస్తుతానికి నా బ్యాండ్ విడ్త్ ప్రాబ్లం వలన.
మీరు ఆడియో ప్రస్తుతానికి మ్యూట్ పెట్టండి, ఇంట్రొడక్షన్ అయిన తర్వాత అప్పుడు ఆడియో ఇచ్చేద్దరు.
అలాగే సార్, మీరుంటే అలాగే. ఓకే, పక్కనే ఉన్నారా మరి చెప్పరే మేడం?
నేను గీతంలో ఉన్నాను మేడం, నేను ఏయూలో వెళ్ళి ఆ భాగ్యం లేదు, నాకు గీతంలో ఉన్నాను.
నో ప్రాబ్లం. ఓకే ఓకే సార్, అయితే మ్యూట్ చేస్తాను మేడం, వచ్చినప్పుడు ఆన్ చేస్తాను. ఓకే.
ఇది మేడం మూర్తి మేడం మేడం
Am I visible, madam?
విజిబుల్ మేడం.
Namaste, sir.
How are you, madam?
Thank you, sir.
మీ గురించి కూర్చున్నాను ఇక్కడ లెక్చర్ మేడం.
Thank you, sir. Thank you, sir.
సో హౌ ఎవ్రీథింగ్ గోయింగ్ ఆన్ వెల్?
Fine, sir. Fine.
మా వాళ్ళని అడిగాను చెప్పండి. మీ వాళ్ళు రిజిస్టర్ చేసుకున్నారా మన దాంట్లో?
Sir?
ఎవరన్నా మన వాళ్ళు రిజిస్టర్ చేసుకున్నారా అక్కడ నుంచి?
గ్రూప్ లో షేర్ చేశాను సార్, చేసుకున్నారు. డిపార్ట్మెంట్ స్టూడెంట్స్ వాళ్ళు ఫ్యాకల్టీ చేసుకున్నారు సార్, షేర్ చేశాను సార్.
రూపా, సుశీల, మాధురి
ఉన్నారు సార్ అందరు. సార్ అడుగుతున్నారు సుశీల, మాధురి అంతా లైన్ లో ఉన్నారు అని పక్కనే ఉన్నారు సార్, నేను స్టాఫ్ రూమ్ లోనే ఉన్నాను. రిజిస్టర్ చేసుకున్నారు సార్. సార్ అడుగుతున్నారు కనిపిస్తారు ఒకసారి చెప్పండి. ఇక్కడే సార్.
స్వరూప, హౌ ఆర్ యు? ఏ సుశీల కనపడలేదు
రండి మేడం.
ఏమంటే ఫార్మర్ ఈసీ మెంబర్ గారు గుర్తున్నా? ఫార్మర్ ఈసీ మెంబర్ గారు గుర్తున్నారా అంటున్నాను. సో థ్యాంక్ యూ ఫర్ రిజిస్టరింగ్. మా ప్రొఫెసర్ చంద్రలక్ష్మి చాలా ఇంట్రెస్టింగ్ గా చేస్తుంది. మారుతి మేడం ఫ్రెండ్. మాధురి రిజిస్టర్ అవ్వలేదా? మాధురి మేడం ఓకే ఓకే అడిగాను చెప్పండి, అందరూ అడిగాను చెప్పండి అమ్మా. రైట్ రైట్, థ్యాంక్ యూ ఫర్ కమింగ్. బాయ్. స్టార్ట్ చేస్తాను.
11 లెక్చర్ స్టార్టింగ్
11:15 స్టార్ట్ చేస్తాం.
11:15 ఓకే ఓకే. గింజర్ అంటే ఎక్కడికి వెళ్ళారు? గింజర్ చంద్రబాబు మీరు
ఆర్గనైజింగ్ సెక్రటరీ.
ఓకే ఓకే.
మారుతి మేడం ఎవరు వచ్చారు ఇప్పుడు?
నవీన మేడం వచ్చారు సార్.
ఓకే ఓకే ఓకే, నవీన్ అడిగాను చెప్పండి.
చెప్తాను సార్.
హ్యాపీ.
నమస్తే సార్, బాగున్నాను. నేను పిన్ చేయలేదు పక్కన.
బాగున్నారా?
బాగున్నారా అని అడుగుతారు, చెప్పండి.
అందరూ ముసుగులు అయిపోయారు.
బాగున్నారా సార్?
బాగున్నా, గుర్తుపట్టలేకపోయా. అందరూ మాస్కులు పెట్టుకుంటూ ఉంటే గుర్తుపట్టలేదు. థ్యాంక్ యూ ఆల్ ఫర్ రిజిస్టరింగ్. I'm thanking all of you for registering అంటున్నా.
Thank you for registering.
Okay, we'll start it.
Yes, sir.
Welcome back to the fourth session. Madam, Maruti madam.
I'm hearing, madam. Please go ahead.
Okay. Welcome back to our fourth session. I welcome Dr. Maruti madam for the 2-day webinar in Trends in Biotechnology. It's my pleased duty to introduce Dr. Maruti madam. Dr. Ellamraju Maruti, presently working as Professor and Chairman, Board of Studies in the Department of Sciences and Biotechnology and in charge Director, Academic Audit Cell, Krishna University. She is having a 20 years of teaching experience. She has qualified UGC SLET in Botany 1998 and acquired PhD from Andhra University. She also got CSIR Senior Research Fellowship in 2000. Dr. Maruti madam got Best Researcher Award from Geetham University in 2011 and published about 78 research papers in national and international journals with total impact factor of 30. She completed research projects funded by UGC and BAR totaling to an amount of 50 lakhs and consultancy projects worth of 1 crore for HPCL. She guided 6 PhD and 80 UG and PG projects. Now I request Professor Maruti Madam to deliver a talk on grey biotechnology, a key to sustainability. Thank you, Madam.
You can present to me.
Now
Good
you
morning,
can,
madam. Yes.
good morning, Madam.
Good morning. Namaste, sir. And I thank all for you first for giving me an opportunity. And thank you, sir. And now I'm going to start my talk and I'm going to share my screen. Please give me one moment. Because of bandwidth problem, I switched off my video. Hope my audio is visible to you. Sorry, audible to you. So the title of my talk is today, Grey Biotechnology, a Key to Sustainability. When I saw the list of the participants, I understood it's heterozygous group like pharmacy, chemical engineering, and biotechnology and biology. So I designed my talk in such a way. First, I'll cover 2 minutes on environmental performance index, then followed by colors of biotechnology, grey biotechnology, applications and recent trends relevant to this title of the webinar. I'm not going to teach any talk about technical details. I'm going to cover a few research discoveries, we say innovative discoveries, uh, which help new researchers as well as few faculty members. They can take up some project works because these are some new topics. I'm not able to prepare PPTs. I will show you videos which are interesting, but I will assure you that my talk is confined to only 25 to 30 minutes. As per recent rankings, India has ranked 168th in Environmental Performance Index survey conducted by Yale and Columbia universities. They considered 32 parameters. Out of that, India ranked 168. If you look at those parameters, I didn't take social as social parameters. If you look at these parameters, health, air quality, sanitary, waste management, all parameters related to environment and health, they all are scored above 100. This says that it reflects us to we have to take up research or we have to update existing research to improve the score on this criteria. So we know that biotechnology encompasses all the branches of life sciences, irrespective of science and technology. As the term biotechnology looks new, but we know the applications and the processes what we have been using are centuries back are dated back techniques, okay, but they are modified. So how environment and biotechnology is classified into different types based on applications. Similarly, some colors are given to those classification of biotechnology. Here you can see in a clockwise direction, this is white biotechnology, which deals with the industrial processes involving microorganisms. Red biotechnology deals with medicine and human health. Blue biotechnology deals with aquatic ecosystems as well as improving aquatic ecosystems. And gray, I will discuss in a couple of minutes. Coming to violet biotechnology, which deals with the law, ethics, and philosophy related to biotechnology. Yellow biotechnology deals with the food and nutrition. Dark biotechnology deals with the bioterrorism and bioterrorism. And coming to next, gold biotechnology, which deals with the bioinformatics integrated with computer science. Brown biotechnology deals with the biotechnological applications of arid and dry land technologies. Green biotechnology deals with applications in eco-friendly or we can say improve agricultural practices. Last but not the least is our gray biotechnology. If you look at this pie chart, you can understand that uses of color biotechs, red for health and medicine, followed by agricultural biotechnology and white biotechnology are gaining more importance compared to gray biotechnology. So, gray biotechnology is defined in different ways, but in one sentence, we can say it is useful to find solutions to environmental problems by understanding the relationship between plants and microorganisms with their environment. In my earlier talk, earlier speakers have covered all this. The Honeyway biotechnology play a key role in protection and maintenance of biodiversity, removal of pollutants and contaminants using microorganisms, and use of plants. Madam covered already in earlier talk. Bioremediation, phytoremediation, biodiversity, microbial ecology. These are one. This slide I'm skipping because in my earlier speaker has covered all these topics in detail. Different techniques employed in phytoremediation, phytodegradation, phytoextraction, phytovolatilization, etc. So coming to the first, and as per my knowledge, first commercial product which was released into the market for bioremediation is oil japper. Oil japper. It was developed by Banwarlal of Terry. He prepared a consortium of five bacteria which can degrade oil. In other terms, which can degrade converts hydrocarbon compounds into CO2 water dead mass, which is environmental friendly. We know oil pollution is a menace for both land as well as aquatic ecosystems, particularly marine ecosystem. This was the first commercially launched compound, oil japper. He called it as also oily vorus. Oily vorus. Five oil companies in India adopted and they are doing. Just I will show 1 minute video. The application of oil japper on oil contaminated sites. So for chemical engineering, as some of people are also attending, a compound was developed which is just like sponge, which can absorb oil and we can reuse it later. Anyway, I'll leave this presentation with the organizers. You can watch it. The next important discovery or research related to uh, environmental biotechnology is Silk Leaf Project. A student from Royal College, he made a leaf which is embedded with chlorophylls and in a silk protein. This is an innovative project actually which can use in reducing carbon emissions. So coming to India, I think on Independence Day of this year, Modi ji announced that a 5 crores women are provided with the sanitary napkins with the minimum price of 1 rupee. As per the present country estimates, 12.3 billion disposable sanitary napkins are disposed of every year, and they are not biodegradable. Because this topic is some sensitive topic, nobody is interested to speak it, but two students of IITR came out with a solution. They manufactured a biodegradable sanitary napkin made up of banana fiber. But here, still, there some research is going on to find out alternate for absorbing biogels. This is also one research area we can take up. So again, one more 1 minute video I'm going to show on 100% biodegradable sanitary pads. Yes. When it comes manufactured by Indian company in Bangalore as a startup, they named it as edible water pots, priced at 2 rupees per 100 ml. In Bangalore, they started a work on workbench projects, how it can be used in hotels, cafes, and replace. I think if any students are there, you can take up this project in your college or university level. Yeah. So another interesting research is going on bioinks. Technical definition I gave in bioinks consists of living cells and biologically active components and biomaterials. Hope this will replace the synthetic materials very soon, and it is gaining importance. South Korea is working on it very well, and few commercial bioinks are already launched in the market, but in India it is in progress.
మేడం
మేడం
ఒకసారి మీ స్క్రీన్ ఒకసారి మళ్ళీ ప్రెజెంట్ చేయరా మేడం?
ఒక్క నిమిషం మేడం,
ఆ,
ఒక్క నిమిషం,
ఓకే
ఒక్క నిమిషం చేస్తాను అండి.
మేడం ఓకే.
1 minute.
మేడం
మేడం చెప్పండి.
మీ స్క్రీన్ షేర్ అవ్వట్లేదు మేడం.
ట్రైన్ అవ్వట్లేదా? ఒక్క నిమిషం మేడం, ఒక్క నిమిషం. నేను మళ్ళీ రీషేర్ చేస్తాను, ఒక్క నిమిషం మేడం.
ఓకే ఓకే.
అంటే ఫస్ట్ నుంచి కనిపించలేదా మేడం?
లేదు మేడం, జస్ట్ ఇప్పుడే ఆ వాటర్ బాటిల్ తర్వాత మేడం.
ఓకే, నేను మళ్ళీ రీషేర్ చేస్తాను, ఒక్క నిమిషం. అంటే ఒకసారి నెట్ ఇక్కడ స్లో అవుతుంది మేడం, అందుకని.
ఓకే
లేదు లేదు, మొత్తం బ్యాటరీ బ్యాకప్ పెట్టండి. సారీ మేడం. ఇక్కడ మనకి ఇది ఎక్కడి నుంచి అయిందో చూసి మేడం ఇప్పుడు
లేదు మేడం.
విజిబిలిటీ రావట్లేదండి?
లేదు రాలేదు మేడం.
ఒక్క నిమిషం, నేను ఒకసారి స్టాప్ చేసి మళ్ళీ షేర్ చేస్తాను. ఇప్పుడు పవర్ పాయింట్ వచ్చింది, నెట్ స్లో అవుతుంది. ఒక్క నిమిషం
మేడం, లేకపోతే నాకు మెయిల్ పెడితే నేను షేర్ చేస్తాను, మీరు ప్రెజెంట్ చేద్దరు మేడం, మీ ఇష్టమే.
ఒక్క నిమిషం మేడం, అది అంటే అందులో కొన్ని స్లైడ్స్ డిలీట్ చేయాలి, నేను చేయలేదు అందుకని. ఒక్క నిమిషం ప్లీజ్, జస్ట్ వన్. సారీ ఫర్ ద ఇన్కన్వీనియన్స్. ఒక్కసారి ఐ షేర్. ఎస్
Only thing is not yet. Just working.
ఇస్ ఇట్ విజిబుల్ నౌ మేడం? కనిపిస్తుంది. ఓకే మేడం, ఒక్క నిమిషం, ఐ కంటిన్యూ. పవర్ పాయింట్ వచ్చింది కదా. So there is one more application of biotechnology. It's a great biotechnology. Direct electron transfer to some methanophilic bacteria may enhance the methane production. It is at very nascent stage. For the sake of anger researchers, I am sharing this slide to you. Later, I will mail to madam. For every slide, I gave the link, the source given below. You can go through that one. And coming to another major issue in India, maybe in yesterday topics also it is covered, waste management. I'm not going to touch any solid waste management, but I'm going to discuss about the poultry litter, etc. But as per the present statistics, it is proved that agriculture is responsible for 10 to 12% of greenhouse gas, with meat, poultry, and dairy farming producing nearly three quarters of that. And as per the economist uh, source, in the last 50 years, the number of people on the planet has doubled, but amount of meat we eat has tripled. This picture may not be clear. Actually, its source is from World Economic Forum. You can see every year 50 billions of chickens are slaughtered for food every year. This figure shows that how many animals are slaughtered and how much waste is generated and how it is increasing by year. So, is there any substitute for meat? Yes, biotechnology provides a possible solution to this. There are two ways. One is the cultivation of animal cells in labs. That means growing real mat in petri dish. Can you imagine this? How we can have a meat without animal? See, that is the amazing application of biotechnology. The meat is uh, grown in such a way that it has all taste and texture similar to beef, pork, or chicken. I will show you one. Okay. So this attempt was made by Harvard John and Paulson School of Engineering and Applied Sciences. They successfully grown rabbit and cow muscle cells on edible gelatin scaffolds that mimic the texture and consistency of meat. So be ready, we are going to have a meat, not from animal. Just see this video. So this is also an interesting topic to take up research. And coming to the one work I carried out relevant to environmental biotechnology. This work we did it in around 2000, between 2008 to 2012. When we took up this work, lot of work has been carried out on degradation of feathers using bacteria. There was a paucity exists regarding degradation of feathers by using fungi. So this project work was funded by UGC. So what we did, so in this project, we collected objectives of this project is to identify keratinophilic fungus from feather dumping sites, to identify which fungus is more potential for degradation of feather and which strain we can use it for commercial purpose. So we covered a few areas of Vishakhapatnam. We collected soil samples for slaughterhouses, meat markets, feather dump sites, sewage slurs, chicken stalls, or polluted beach sands and poultry farms, etc. Totally, we identified 25, but out of that, very few are keratinolytic in nature because few fungi are keratinophilic. They love to grow on will not able to utilize or degrade keratin completely. So we in our laboratory by using keratinolytic assays and we measured different cysteine and cystine release, and these are the few photographs which are showing degradation of feather 20 days, 40 days and 60 days, and the potent Chrysosporium troglodytes, Microsporium canis, Microsporium gypsum, and trichophyta and mentagophytes as few are dermatophytes. But what we observed in our study, alkalinity at pH, the dermatophytic tendency has come down in selected fungal strains, and these are the different fungi we observed and we published it. And this is one slide showing the distribution of these fungi from poultry farm soils. And what we concluded in this study, actually the Microsporium gypsum and Chrysosporium indicum both are very good at degradation. We tried one small study also by collecting the assay mixer, and we tested for seed germination tests, and successfully we got 70 to 80% seed are germinated. But in this, after this, we have this work has more if we can take up it at molecular level and to develop processing reactors at industrial level and production of valuable derivatives. Another work, this work we did it in 2010 to 2013 by taking green chili and clustered bean or chickadee. We inoculate them with the selected AM fungi, and we studied the growth response of these plants. And similarly, we measured pre and post inoculated mycorrhizal soils nutrient contents, and we observed phosphorus is growing and plant growth is very promising. This slide shows the this is in cluster bean how the AM fungi are inoculated into the roots. Our study elucidates that in cluster bean, the more number of bonds observed in SDS phase when they are inoculated with the consortium of mycorrhizae with rhizobium. These are few pictures. Okay, so what we recommend AM fungi association with consortium in mineralization pathways, its role should be identified. I told you a couple of minutes before. Molecular approach and adaptation process rhizobium along with AM fungi under field conditions is the need of the hour. Of course, this is the recent one. There's already Terry has a division of AM fungi. They have started focuses on conserved protein encoding, housekeeping, as well as functional symbiotic associate genes of these fungi, which can serve as molecular trait indicators and may open opportunities to develop agriculturally important AMF isolates for sustainable agriculture and industrial and environmental management for rapidly changing climate adaptability and production. This is very small work funded by HAPCOST in 2000. All of you are well aware of the mushrooms. So mushrooms generally are cultivated by using rice straw as the substrate. So in this study, what we did, there is a common 50% of rice straw and 50% of leafy vegetable waste dried used as a substrate. The results are promising. We did it in 2000, funded by AppCost in our laboratory. This is also very good work. I think now we tried with leafy vegetables. If any young researchers are there, students are there, you can take up with the other combination of the vegetable waste, and you can give hands-on training to some rural people. These are I'm skipping. Madam, can you hear me?
Yes, madam. Cheppan, madam.
Madam, this was only I'm having. I think I tried to relate few important issues which can help to researchers as well as which are going on in present scenario relevant to biotechnology, because your webinar title is Friends in Biotechnology. I took one example for environment, oil japper, and one for medical and health. Another is to cut greenhouse emissions that alternate meat. Thank you, madam. Let me know any queries or questions are there.
Any queries? Any queries from the participants?
Hello, ma'am.
Yes.
Ma'am, we should grow like meat.
Yeah, madam, it is still not completed.
No, so what do you think about plant-based meat? Like there are companies that making it. Hello.
Hello.
Yeah, I'm saying, what do you think about plant-based meat? Why can't we go for that plant-based dairy?
That's what madam I'm saying. That is another research challenge for us. We have to think of it. So they are trying to grow using animal cells as a source of meat. We have to think of that. That is the research challenge ahead of us.
No, growing cells of animals, it is in research phase, but plant-based meat, plant-based dairy are in market, right?
Yeah.
Like Beyond Burger and Impossible Burgers are there in US right now. I don't think it's in India.
Yeah. That's what I'm saying. See, these are under trials and some are maybe about to launch, but similarly, like I told you, water pods, we have to take up in India also as a startups or any through research projects.
So what do you think is more like, has more, what should I say? More scope in future, like the production of cells or plant-based?
So it has to, madam, but unless we will come out with a product and we have to see the product quality, then only we can. This also good question. So, sometimes in some countries they are providing
Sorry, ma'am, I can't hear you. Sorry. Can you pardon?
Okay. Hello.
Yeah.
Madam, maybe that is also an option, madam. We have to try.
Thank you.
Okay. Madam?
Madam.
Thank you, madam. So I think I have not seen any.
Excuse me, ma'am.
Hello. Yes.
Excuse me, ma'am.
Yeah.
Ma'am, regarding this bioprinting, I have a small doubt. I mean, like a small query. As we know, these the guts we have. These are not regenerating themselves once they got degenerated. Unlike epithelial cells, epithelial tissue, these gums cannot be regenerated. As I said, this tissue engineering falls under bioprinting only. With the help of tissue engineering, can we solve this?
So I think I read it somewhere, but I'm not sure. They're trying with stem cells. I read it somewhere, but I'm not sure which. So they're trying with stem cells. I read it somewhere. I'm not sure.
Hello,
Hello, can you hear me? Hello.
ma'am. Yes, ma'am. Yes, ma'am. I can hear you. Yes, ma'am.
Yeah. That's what your question is absolutely right. But I read it somewhere. They're trying with stem cells, but I'm not sure of that. When I'm going through this, I heard about it. They're going for stem cells. They're trying with.
Okay. Okay. And regarding this bioprinting, we can print any tissue. I mean, like skin, a part of skin
Yeah,
or
that's
a
what
face
they'll
tissue.
know in future. You can replace your skin or any organ. It
Yes,
is
ma'am.
under trial. That is the
Yeah, ma'am.
future benefits, or I can say application.
How can this? I mean, like, how can we make it most ethical way? I mean, like, this can
Sir,
lead
but
to.
I do understand because see, in the beginning of my talk, I told you there is one violet biotechnology which deals with law, ethics, and philosophy. So that
Yes,
deals with that one. Yes, not only that. Regarding meat also, again, it should meet with the ethical. So
yes,
these
ma'am.
should be solved. So these are that's what we have to work on it.
Oh, okay, ma'am. Thank you, ma'am.
Okay.
That's all.
Any more questions from participants?
Master, thank you, madam. Then.
Thank you, madam. Nice
Namaste,
presentation.
sir. Thank you, sir.
Thank
But
you.
I'm not expert in PPT. Kindly bear me. Sorry. My size
So if there are no questions, we wind up this program. Chandana,
Sure, sir.
close it.
Thank you, sir. Madam, I'm switching off my thing.
1 minute, madam.
Madam, okay, 2 minutes, madam.
Okay.
Thank you, madam, for your lecture. I can say definitely your lecture is very useful to all of us students.
Thank you, sir.
Now, I would like to close this two-day webinar on Defense and Biotechnology by saying my word of thanks. It's my pleasant duty to formal word of thanks. I express my profound gratitude to all the experts for their invaluable insights into various aspects of biotechnology they offer to us. We are all grateful to them. The person who is instrumental in organizing this webinar is our head of the department, Professor Pulipati sir. We are thankful to our dynamic principal, Professor P. Srinivas sir, who is always ready to support and guide us. I'm also thankful to Professor C.H. VVS Bhaskar Reddy sir, Nodal Officer, TechVip Phase 3, for financial support, and Professor K. Ramasuda, Honorary Director, Computer Center, and their technical staff for giving us a technical support. I'm also thankful to Professor D. Apollanaidu sir for his engagement. We are all thankful to distinguished invitees, heads of the institutions, heads of academic departments, faculty and supporting staff, students of the university for making this occasion a grand success. Thank you, Vanessa.
Madam, may I also say, I was also there in the meeting, madam.
Sir.
Professor King is there.
Namaste, sir.
Namaskaram.
Namaste. How are you, sir?
Just listening to these lectures,
Yes, sir.
very
Thank
nice.
you.
This is a very good, exposition for the students and also the research scholars. I thank department and especially King Garu and the organizer, madam. Thank you so much. I was also benefited by this. Okay.
Sir,
Thank
sir.
you.
Thank you very much, sir. Thank you, sir.
Thank you, sir.
Okay.
Madam? Madam, are you there?
Which I'm going to share in the chat box and also in the WhatsApp group.